So this is the murine sacculus or sac. So let's again zoom in a small part. This is the peptidoglycan. We know it now. It has many identical subunits of disaccharide and tetrapeptide. Peptidoglycan. Now what is glycan? Glycan is the compounds where large number of monosaccharides are linked with glycosidic linkages. So coming to the glycan part, that is the sugar backbone. This is the sugar derivatives we know, and these are the glycans, means glycans are the sugar backbones. Now, two types of molecules are there. One is N-acetyl muramic acid and another is the N-acetyl glucosamine. Means suppose this is the NAG N-acetyl glucosamine. NAG means N-acetyl glucosamine, and other one is the NAM molecule, that is the N-acetyl muramic acid. This is the NAM molecule, means N-acetyl muramic acid. These forms the sugar backbone. Now these glycans let's draw another layer, two consecutive layers. These glycans are combined with different amino acids. Actually, there are set of identical tetrapeptide attached to the NAM molecule. Short peptides of 4 amino acids arranged connected to the carboxyl group of the NAM molecule. These are attached with the carboxyl group of the NAM molecule. Like this. Specific patterns, specific amino acids arranged in specific pattern. So, what are those amino acids? Those you can remember with the mnemonic AGLA. A-G-L-A. Now, A for alanine, G for glutamine, A for alanine, G for glutamine, L for lysine, and again A for alanine. G for glutamine, remember, not glycine. Glycine is the pentaglycine bridge. So this is, you can remember it like this. So this is not present in the tetrapeptide side chain. There is glutamine in the tetrapeptide side chain. And another important thing is, this is LD-LD. Starting with L, D, L, D. If you even forget whether it is starting from L or D, you can remember that's L for L. In lysine, this is L. So then you can calculate it as LD, LD. So L alanine, D glutamine, L lysine, and again D alanine. These are the short tetrapeptide chains connected with the carboxyl group of the NAM molecule. These are the two layers of the glycopeptides, peptidoglycan. Now, how are they linked? So that they cannot be separated from each other and so that they form a single gigantic molecule or say continuous sheet of layers. Third position of this amino acid, they will be connected with the fourth position of the next fourth position of the tetrapeptide side chain of the next layer. So they are holding their hands like this. 4 and there will be another 4. So third position of one layer with fourth position of the next one. Third position with the fourth position. And similarly in the next layer again third position with the fourth position. And these NAM and NAG molecules are attached among themselves with 1,4 beta glycosidic linkages. This is the 1,4 beta glycosidic linkage. This is between NAM and NAG molecule. So now this third position, this is important. This third position is the most variable position. So that means this L-lysine, this is the most variable position. Because in gram-positive bacteria, the third position contains L-lysine. Bacteria is gram-positive or gram-negative. That is due to difference in their cell wall structure again. So here lies the importance of cell wall from our perspective for identification. We will come to this later. So in gram-positive bacteria, the third position of the short tetrapeptide chain, there is L-lysine, but in gram-negative, this will be diamino pimelic acid, DMPA. This DAPM, sorry, DAPA, diamino pimelic acid. So, for gram-positive bacteria, how will be the cross linkage? This will be arranged the short tetrapeptide chain. I am talking about the short tetrapeptide chains connection between them. This third position L-lysine will be attached with the next D-alanine of the next short tetrapeptide chain of the next series of NAM molecule attached consecutive layer. And in gram-negative bacteria, diaminopimelic acid that is present in the third position of the peptide chain will be attached with the D-alanine of the next consecutive tetrapeptide chain. And again L-lysine of that chain will be attached with the D-alanine of the next peptide chain. And here again DAPA of one chain to the next D-alanine of the next tetrapeptide chain. And what is this cross bridge? This cross bridge is nothing but this is pentaglycine bridge. Pentaglycine bridge, and these are identical. These are identical pentaglycine cross bridge. Identical pentaglycine cross bridge. This backbone is same in all bacterial species. The sugar derivative, the backbone, in total, but tetrapeptide side chain and pentaglycine bridge vary from species to species. Tetrapeptide side chain as well as pentaglycine bridge, those are identical in a single species but vary in in different species. So in gram-positive bacteria, this network of single gigantic molecule of peptidoglycan or murin saccharols, this is thick 50 to 100 layers, whereas this is 50 to 100 layers thick. Whereas in gram-negative bacteria, this is very thin and this is just 1 to 2 layers. 1 to 2 layers. And this is 60 to 80 nanometer and this is only 1 to 2 nanometer thickness. Cell wall accounts for 20 to 30% of dry weight of cell. Bacterial cell wall accounts for 20 to 30% of dry weight of cell. So, I will discuss the differences between gram-positive and gram-negative bacterial cell wall in some other videos. Gram-positive bacterial cell wall has peptidoglycan and covalently attached techoic acid. So, in gram-positive bacterial cell wall, peptidoglycan plus techoic acid, and this is covalent bond between them. The techoic acid is very important. Techoic or techoic acid.